Hello, this is class Bio 225, Genetics from Genes to Genomes with Dr. Lucy Abgaryan. Today we are going to cover Chapter 7, Part 2. During last class, we have talked about different mechanisms of DNA mutations. Today we are going to talk about mechanisms that cells apply in order to either avoid these mutations or fix these mutations. Let's start this from stating that lucky for us, in bacteria and in humans, the chances of mutations are very, very low. It's 1 in every 10 billion. So, however, it still occurs, and there are two ways that this can be minimized. This mistake can be minimized. One of them is proofreading. It's the function that the DNA polymerase has. It's very important to understand that the DNA polymerase, it's a complex protein. It has different components that are responsible for different functions. So what it does, if you remember probably from previous classes, that it goes from 5 to 3, adding the complementary pair to original parental DNA. However, after certain fragment, the part of the polymerase, which is called exonuclease, will go back from 3 to 5 and recognize it, and if there is some mismatches, it will exercise those. And the second method for the minimizing the mistakes is called methyl-directed mismatch repair. That is the ingenious property of cells to be able to differentiate the parental DNA, the original DNA strand, from the newly constructed DNA strand. So what happens is that DNA cells have a property of adding methyl group to DNA after a certain amount of time. So which means that the new strands will not have that methylation, which will be a place for cells to recognize if this is methylated DNA, that means that's the original one, that means that's the correct version of the sequence, and if there is a mismatch, then the one that doesn't have the methyl group, that would be the one that is the wrong one. So that's the one to avoid. So let's look at the graphical representation, very simplified model actually, of proofreading function here. And so this is what's happening, what the exonuclease does by going through 3' to 5'. It will recognize the mutated base pair, which will be mismatching, and it will cut the phosphodiesterase bond and replace that mutated base by the correct one, and the DNA will continue building on after that point on. It is important to note that if there is a mutation, so for here, for example, instead of cytosine, on front of guanine, there is adenine, which means that their hydrogen bonds are not going to be high energy. It's going to be very unstable structure. So there will the energy of the bond is not going to be as high, and the system is not going to be as stable. So cutting this is not going to be difficult. It's not going to require high energies. So it's not 100% going to improve the proofreading function, the DNA mutations. However, this mechanism improves fidelity of replication by 100-fold, which means if we did not have this, we were going to have mutation instead of 1 in every 1 billion. Now we would have it 1 for every 10 million. So I think this is still pretty significant and very important mechanism for avoiding mutations. Unlike the previous mutation mechanism, crossing over is much severe mutation, which is going to have much severe and serious impact on a system. So let's look at an example. And if you remember, we have talked about crossover mutations, and this is something that happens during meiosis. We have talked about it during previous couple different classes. And so here are two genes that are aligned, two homolog chromosomes, and let's say within a system they are not aligned properly. So as you can see, the second part, the red is aligned with green, so they are misaligned. And what will happen in this case, the crossover will take place, and the outcome would be that we will have one gene with deletion, which is missing full yellow, full red, or full green part of the gene. And we have a duplication case, the bottom gene, where we have complete red, we have complete green, and also we have additional hybrid gene that is partially green and partially red. This is going to have more severe impact on the system in general than the previous point mutations that we have discussed. Another type of mutation is the transposable one, where part of the gene can be either cut completely from the sequence or duplicated and transposed into new DNA sequence. And those parts are called transposable elements. And here is the graphical representation of how that is, the model, how that is taking place. Let's talk about the experiment that was conducted by Miller to see the mutations that are caused by environmental factors such as X-ray, for example. So the scientists designed an experiment where they were trying to understand the extent of the effect of X-ray radiation that are above the natural occurring levels on Drosophila. And they were doing that by detecting the X chromosomes because it's easier to detect X chromosomes. So whichever ones were dying, they were the ones that were affected and had the mutations, and ones that didn't, the survival ones were the ones who were not affected by the radiation. So here, let's look at the model of this experiment. So there is a male that was X-ray radiated, and there is a female that carries Bar gene, which is X-linked gene. So their offspring will have, which is the Bar female. So let's look at the two scenarios. If the F1 female is mated with wild type male, it will cause into having either offspring that will carry the mutation, so it will die, or it will have a offspring that will not have the mutation. It will and it will survive, the one with bari. In case if the female has no X-ray induced recessive lethal on X, then both offsprings will survive. Just one will have bari and another will not have the bari. So now let's talk about chemical mutagens. So there are forms of bases that are very close. They are analogs to the original bases. For example, the 5-bromuracil, for example, it is being complementary to adenine. It can be substitution for thymine. However, its deprotonated form is being able to stably bond with guanine. So let's look at the bottom scheme where there is a DNA with adenine thymine, and when we have a switch to the chemical analog where we are dealing with bromuracil. So there is a bromuracil instead of thymine, complementary paired with adenine. So when we have a replication process, and if the transformation of the bromuracil to its more rare deprotonated form is taking place, then after the replication process, we will end up having cytosine, because that's what the complementary pair for guanine is, and it will cause thymine to cytosine point mutation. So that is one case of chemical mutagens. Another chemical that is causing chemical mutagenesis is hydroxyamine, where the hydroxyamine is the agent that is causing the hydroxylation that adds OH group to cytosine, and it makes hydroxycytosine, which is actually complementary to adenine. So cytosine is complementary to guanine. However, the hydroxycytosine is complementary to adenine. So if we have that mutation and then we undergo the replication process, then instead of and we have the transformation of cytosine to hydroxycytosine, then it will create a complementary pair with adenine, and then on the end of the replication process we will end up having adenine thymine instead of guanine and cytosine. So this is another chemical mutagenesis process. Another group of chemicals that can cause chemical mutagens is the alkylating agents. For example, the ethyl methane sulfate. So this agent is causing transformation of guanine to ethyl guanine, and now the ethyl guanine, instead of binding to cytosine complementarily, it will create a hydrogen bonds with thymine. So again, undergoing the replication process, so now instead of guanine cytosine complementary pair, we will end up having adenine thymine complementary pair. Yet another agent is the deaminating agents, and they are causing of cytosine to uracil changes and adenine to hypoxanthine changes. So obviously this is happening because those two bases are the ones that have the amine group, so the amination will take place with those two, and also the cytosine to uracil, we have talked about it previously, that the amination will cause this transformation, and then end of the day uracil is being the complementary pair for adenine, and the interesting fact is that poxanthine is complementary binding to cytosine. Here are the schematics below for both of the cases, and in case of cytosine deamination, we are having cytosine guanine to thymine adenine mutation, and in case of adenine deamination, we are having adenine thymine to guanine cytosine mutation. Finally, the last one that we are going to talk about is the intercalating agents that are causing chemical mutagenesis. So what is intercalating agents? Those are molecules that can get between strands of DNA and basically create a lesion. And when there is a lesion during the replication process, any random complementary pair can come and get inside between these two lesion sequences, and after that the entire sequence from that point on is having a gene shift. One of the agents that is intercalating is proflavin, for example. And there are other agents that can have the same property. So next time we will pick up from talking more in depth about mechanisms that are very accurate for DNA repair of all of above mentioned mutations.